So today I'm really happy to share some unpublished work related to our efforts in understanding macrophage heterogeneity. So the historical perspective of a macrophage is that bone marrow monocytes enter the bloodstream, and then these blood monocytes differentiate into tissue macrophages. However, we now know that it really isn't this simple, as not all macrophages are derived from adult monocytes. In fact, there are several origins, including the embryonic yolk sac, fetal monocytes, and adult monocytes. So this presents the first layer of complexity related to macrophage heterogeneity. So the question is, where do we start our study of macrophage biology? Well, for a long time, macrophages were studied as mixed populations, but we've come a long way with single-cell technologies, which shows that even within an individual organ, there are many transcriptionally heterogeneous subpopulations that exist. However, this raises yet another layer of complexity. How are these different subsets in different organs related to each other, if at all? And this is further complicated when you consider the number of different studies with different approaches and also different species. So we do appreciate this heterogeneity, but there's a lack of a consistent approach that's limiting our ability to translate findings from one study to another. So the question that motivated our research is there an underlying macrophage organization that can inform a common starting point to study tissue macrophage heterogeneity and biology? And so we focused on commonalities between 5 organs in the mouse and human. And although there exists major tissue-specific differences, there is a very small gene profile for 3 macrophage subsets that was consistent across organs. And this transcriptional organization lent itself to track with common biological properties. So today I will be walking through the data that helped us build this common starting point in macrophage biology. So we focused on the heart, liver, lung, kidney, and brain, and we sorted total macrophages using the marker CD45, and we casted a wide net for CD64 and CD11b to ensure that we capture all macrophages and perform single-cell RNA sequencing. So starting with the single-cell analysis of mouse heart macrophages at the steady state, where we excluded monocyte and dendritic cells, we see at least 3 subpopulations of cardiac macrophages. The first subset, over here on the right in gold, expresses the genes TIMD4, LIVE1, and folate receptor 2. So for short, we term this subset TLF macrophages. The second subset in green on the other side lacked expression of subset-defining genes of TLF macrophages, but it expressed CCR2. We term this subset CCR2 macrophages. We also found a third subset in the middle here. It didn't express the subset-defining genes of the other groups. However, it highly expressed antigen presentation genes similar to CCR2 macrophages, yet it lacked CCR2. So this likely represents a more generalized population of macrophages, and for simplicity, we've termed them MHC2 macrophages. So our goal was to assess whether a similar substructure can be identified in other organs as well. So in the lung, we have this very dominant population of alveolar macrophages, but also a smaller group of interstitial macrophages, which was our interest. So zooming into these interstitial macrophages, we find at least 3 subsets that track with the reciprocal expression of TIMD4 and CCR2, and a group here in the middle that lacks both of those genes. In the brain, we have the highly tissue-specialized microglia, but also a group here termed BAMs, which stands for border-associated macrophages. And again, zooming into these BAMs, we see at least 3 subpopulations, again, tracking with reciprocal expression of TIMD4 and CCR2. This pattern is extended to the liver, where the TLF macrophages are actually the very well-studied Kupffer cells, which express the gene CLEC4F, and also extended to the kidney, where the major population are the CCR2 macrophages, and the TLF macrophages are relatively minor, yet still present. So at this very broad level, we can see that TIMD4 and CCR2 expression tracks with 3 subpopulations in 5 organs. But we wondered, beyond these couple markers, how similar are these macrophage subpopulations to each other? So to answer this question, we looked at differentially expressed genes that are found in each subset in each organ, and then we compare them across organ to assess the degree of overlap. So starting with the TLF macrophages, we see that about 75% of the subset gene signature is organ-specific. This means that those genes were expressed in one organ only, and only 11% of the genes were shared in at least 3 organs. CCR2 macrophages had a smaller percentage that is shared in 3 organs, and MHC2 macrophages appear to be the most plastic or responsive to their tissue, as only 4% of the genes were shared in 3 or more organs. Now, what's very evident here is that while we attempted to look for commonalities, the greatest signal was in fact differences, and there's only a small aspect that is similar. So we defined a core gene profile for each subset that's composed of genes that define the macrophage subset in 3 or more organs, shown here with their expression levels. So we see here that a small gene set is consistent for each subset in each organ. We then focused on the 4 top cell surface markers, TIMD4, LIVE1, folate receptor 2, and CCR2, and using random forest modeling, we showed that the expression level of just these 4 genes is sufficient to predict our macrophage classification with high accuracy in all organs. So we've really narrowed it down to just a few predictive genes. So then the next critical question was, while single cell provided us with the macrophage classification with a small conserved aspect, we wondered whether it can also track with common biological properties. So the biological work in the next 2 slides was performed by a graduate student and postdoc in the lab. So we used the top cell surface markers identified in single cell, TIMD4 and CCR2, and we assessed the macrophage subpopulations at the protein level by flow cytometry. And starting with the liver, we see that stratification using TIMD4 and CCR2 reveals 3 subsets, which expressed either TIMD4 or CCR2 or neither. And this was observed in all 5 organs, where we see that the TLF macrophages, like single cell, co-expressed LIVE1 and folate receptor 2, whereas both of the other subsets highly express antigen presentation genes and can be distinguished by the expression of CCR2. So we then use this stratification to assess if these 3 subsets differ biologically. Specifically, we assess the relationship of these subpopulations to monocytes. We use parabiosis where 2 mice are joined and they share their circulatory system. So this means that monocytes from one mouse can enter tissues in the other and differentiate into macrophages. So we first paired 2 wild-type mice together for a short-term 5-week-long time period. And after this short time period, we see that CCR2 macrophages have a very distinct life cycle, as nearly all CCR2 macrophages were donor-derived, meaning that they were rapidly replaced by monocytes. And this is very interesting because CCR2 macrophages have this very distinct life cycle, and this is consistent in every organ that we assessed. So there seems to be less replacement of MHC2 macrophages and no replacement of the TLF macrophages in this short time period. So we then assessed longer time periods, and we ramped up chimerism by pairing CCR2 deficient mice with wild-type mice and focusing on the TLF and MHC2 macrophages after 6 and 10 months. We see that there's slow replacement of both the MHC2 and TLF macrophages, and interestingly, when monocytes do replace macrophages, they do so to a finite maximum, where there's no additional replacement at 10 months in comparison to 6. We observe these very interesting patterns conserved across organs, but there are also some tissue-specific responses. For example, in the liver, the MHC2 macrophages actually do have more monocyte contribution at 10 months, and in the kidney, there is very little replacement to both subsets. So the key takeaway here is that the single cell informed a small gene profile that also tracked with conserved life cycles across organs. So I'll very quickly show some data in the human tissue that correlates with what we observe in mouse. So in human cardiac tissue in both the neonate and adolescent, at a broad level, we see at least 3 subpopulations, where the first tracks with genes in the mouse TLF macrophages, such as LIVE1. The second one tracks with the gene CCR2, and the third one expresses neither, but expresses high levels of MHC2. So this was consistent in publicly available data of the human liver, the human lung, and the human kidney. However, we wanted to assess beyond these core markers how similar are human macrophages to those in mice. So we performed a more formal analysis using Garnet, which was published recently in the lab of Dr. Cole Trapnell. So the Garnet algorithm trains a classifier based on a cell type specific signature and then scores cells in a different data set. So we generated a signature for each subset in the mouse and scored the human macrophages, and we observed that human macrophage 1 was most similar to mouse TLF macrophages, whereas human macrophage 2 and 3 were less specific, as they were both enriched in the mouse CCR2 signature. So looking closer at the TLF macrophages versus human macrophage 1, we found that around 20 to 55% of the genes were shared, meaning that they're enriched in this subset in both the mouse and the human. So we show that TLF macrophages are the most conserved subset across species. And this is very interesting given the lack of conservation between mice and human previously reported. So to summarize, our goal was to attempt to simplify the complexity that comes with macrophage heterogeneity and to understand organizing principles that may exist. But we found that this was a very complex undertaking and that there are major differences between organs. However, there does exist a small core gene profile that tracks with 3 macrophage subsets in 5 organs. And this profile tracked with a common relationship to monocytes across organs. We hope that our work provides a starting point for those who study macrophage biology and a resource to understand macrophage heterogeneity in a range of fields. I'll end by thanking members of our lab, and in particular, the 3 highlighted here, who are the co-lead authors of this study, and our supervisor, Dr. Slava Eppelman, for mentorship during this complex undertaking and for introducing me to computational work, which has brought me here today. I'm happy to take any questions.
Thank you very much, Amira, for a really cool talk. We have time for a couple questions. So Tim Herpelink asks, thank you for a very interesting talk. Have you looked at how your 3 subpopulations correlate to the classical M0, M1, and M2 phenotype classification?
Yeah, so that's a very interesting question. So it has been shown that the M1, M2 phenotypes, they don't completely track with origins and life cycles, in that some of the genes that you see in the alternatively activated M2 macrophages, they do track with our TLF macrophages, so the TIMD4 live1 expressing subset. And some of the genes where the M1 macrophages are more inflammatory, where we see CCR2 macrophages are more inflammatory. However, this is very dynamic in that these macrophage subsets with different origins and life cycles, they can change in the context of disease, and they can become more inflammatory, they can become more reparative. And so we would rather propose this framework that actually tracks with biological properties, where this single cell identified a relationship, identified a gene signature that then correlates with macrophage subsets that tracks with their embryonic or adult origin and also their relationship to monocytes. And so this is a better framework to then assess macrophage function as opposed to an M1, M2.
Okay, great. Maybe let's take one more question. That was a really nice talk, Umira. There's a question about injury from Samar Tej. Have you observed if upon injury in different tissues, these macrophage modules, CCR2 versus MHC2, replace the tissue niche and enforce differential TRM programs conserved across tissues?
So we did not assess conservation in the subsets in the setting of injury, and that all the work was performed at the steady state. However, our own lab has shown in the setting of injury the distinct roles of the embryonic derived TLF macrophages versus the monocyte recruited CCR2 inflammatory macrophages. For example, in the heart, we know that the embryonic derived ones are more reparative. And so we've now extended sort of this framework to 4 other organs and proposed this as a way for other labs who study disease in other organs to assess the distinct roles of these embryonic versus monocyte-derived macrophages.
Great. Thank you very much.